Alright friends, we've got section 6.4c, this time talking about lysosomes and vacuoles. We're going to start with lysosomes as our first topic, and lysosomes would be a vesicle that has digestive enzymes in it. So here's a lysosome, and a lysosome would be produced by the endomembrane system, so this would probably bud off of the trans side of the Golgi, the trans face of the Golgi bodies, and has these digestive enzymes. Specifically, this word hydrolytic, they are hydrolytic enzymes, which simply means they're good at doing hydrolysis, so they break things down. So they have these enzymes, hydrolytic enzymes, and there's two different scenarios here where this could be used. So one's here, the other one down here. One scenario would be if you have some form of a cell in humans, like a white blood cell. White blood cells go around and they engulf bacteria. Excuse me, just burped there. Anyway, so they're engulfing something here, and so you've got a brand new set of particles. If it was a amoeba, this might actually be food. If it's a white blood cell, it's actually a bacteria that we're going to try to destroy. But either way, something needs to be digested. That food, as it comes in there, or bacteria, it's held in a membrane, part of what used to be the plasma membrane of the cell, and we call that a vacuole. Then it's a storage chamber, but a food vacuole. And all that happens is the enzyme is going to join that vacuole, and they're both made of membranes, the same kind of membranes, so we can just fuse the membranes together. It's going to make a slightly larger vacuole here, but those digestive enzymes can then break down whatever it was, and if it was some kind of protein that's being broken down, it'll actually break it down to its amino acids, and then those amino acids can pass through the membrane here and go into the cell and be used to build different proteins. So that is how a lysosome works in the case of digesting food. Lysosomes can also be used down here in autophagy or autophagy, depending on how you say it, where this particular cell has taken a mitochondria that is probably damaged or something like that, taken that mitochondria and surrounded it by a membrane, so we have a vesicle with a damaged mitochondria, and our lysosome is going to come and join with that, so they're going to work together, join together, and we're going to end up digesting the mitochondria. That way we can break down the broken organelle and use those pieces to make something else. Okay, so that is lysosomes. The reason I threw this video in here is one of your assignments actually asks you to explain a lysosome being made, so and what it does. So this is what it does. Next slide here, we're going to talk about vacuoles. Okay, here we have vacuoles as our topic, and vacuoles is basically a storage compartment. That's the best I can say. It's a storage compartment. There are all types of different vacuoles storing different things, and so there's three ones that were listed, and we're just going to kind of talk through those. There are food vacuoles, which we actually talked about on the previous page, where if something engulfs food and that food is then surrounded by a membrane, they'll call that a food vacuole. So that was that one. If we look at a contractile vacuole, okay, contractile vacuoles are found in protists, and protists basically live in fluid environments, liquid environments, and so down here we have, this is not a paramecium, but it's something, a ciliate that is similar to a paramecium, but it's a single-celled organism that lives in water, and because it lives in pure water, most of the time, or this one lives in pure water, as you learn about diffusion and osmosis, this will make more sense. There would be water, H2O, constantly, that's a horrible writing there, constantly flowing into this thing, to the point that water would just keep flowing in and keep flowing in until this thing would burst. So they actually have vacuoles that take that water, and this line is pointing to a vacuole, take that water and store it into a vacuole so that the rest of the cell isn't getting larger and larger. So this vacuole would get larger and larger and larger, and then it would contract and it would actually squirt the water out of this thing. And this one happens to have two contractile vacuoles. There's one over here, and you can actually watch them grow. If you have a living paramecium, you can watch them grow as they fill with water, and then they'll squirt the water out, but it's storing water to eventually squirt it out of there. So that's a contractile vacuole. And then the central vacuole is the one you're going to hear the most about as we move forward through the year. A central vacuole is basically just found in fungus and plants, and they can store anything. Okay, and they have multiple jobs, but one thing it can do is if you have a situation where a lot of water is flowing into a plant cell or a fungus cell, a lot of times the cell will store that water in this vacuole that would be in the very center. Okay, this central vacuole. It's just a place to keep that liquid. Okay. Also, if toxins are building up inside of a cell, this contractile vacuole doesn't have anything important inside of it. So if you have some toxins building up, you can actually store those toxins in here, and it keeps them away from all the organelles that are important. Other things you can do, if the cell wants to store some type of molecule for later on, you can store it in this contractile vacuole. And then the last one that kind of goes with what you guys are learning right now is plant cells can actually get relatively large, because they can fill the center of themselves with this big space that doesn't need nutrition and doesn't need to get rid of waste. And so what they end up doing is the usable volume inside of the plant cell, they can make a big contractile vacuole. And so all of this space in red, it's okay if it doesn't get anything through diffusion or food. Okay, the stuff that needs diffusion in order to get food and get rid of waste is all out on the outside here, and it's within a close shot. So it's a way that plant cells can get large and not experience the problems with surface area to volume, because a large chunk of their volume is kind of useless. Is there. And okay, that's it. Lysosomes, vacuoles. That was this video, and you guys have a good day.